HIV, tuberculosis, malaria, and other diseases causes tens of millions of deaths every year. In developing countries, people cannot access the correct cure in time. This happens because proper diagnostic tools are not available. They are expensive and they require a strong expertise. This problem needs a solution. Hi, we are Gabriele, Tommaso and Livio, and we are developing a low-cost, easy-to-use real-time PCR thermocycler. PCR stands for polymerase chain reaction. It's a reaction that replicates the quantity of DNA in a sample through temperature cycles. PCR is a useful piece of diagnostic technology. For example, you can take a sample, put it in the thermocycler and try to replicate the DNA of a certain virus. If the procedure ends with an exponential growth of the quantity of DNA in the test tube, then you have found a virus. Otherwise, if you end up with almost the same DNA quantity, it means that there was no virus in the sample.
EasyPCR is a real-time PCR machine. It is possible to perform the reactions and get the results without any further steps. EasyPCR is accessible because it is low cost. We are changing the real-time technology. Low power. It is portable and can be powered through a battery. And user-friendly. Everyone can use EasyPCR. It is controlled through a Wi-Fi connection, and thanks to an app, you can set and control the status of the reaction. Moreover, you can share what you've done with the community. There are a few low-cost PCR machines in the market, but they are not real-time nor low-power, because they use the same technology that has been used since PCR was devised.
We want to change the way everybody sees biology. We believe that lowering the access barrier to this science will bring about a strong growth in the research. This is what happened in the past 10 years to electronics. We were able to work and follow our passion because we could afford materials and tools. Our team consists of 3 founders, Gabriele Borelli, Livio Saracino and Tommaso Zanotti. We are currently working full-time on the project. We come from electronics bioengineering and have a strong background in the technical field.
The global PCR market is valued around $8 billion and it's expected to grow up to $10 billion by 2021. Initially, we plan to reach schools, universities and biohacker communities. The number of PCR thermocyclers in schools and universities is very limited. Often, students can perform very few reactions during their studies.
Basically, nowadays, knowledge is everywhere. It's so widely accessible that people are reading a lot and they get interested in science and they come up with ideas that they would like to test, but they cannot because they don't have access to the proper technology, the laboratory tools and devices that people use in the lab to test these ideas. But if they had, that would be a very great revolution in how we make science, because science will not be relying only to the contribution of a small population, but by opening up to the contribution of everybody, eventually this is going to lead to some big jump in scientific knowledge.
Biohackers are people passionate in biology, who perform analysis and experiment in autonomy. Many biohacker spaces, called biolabs, are sprouting. Also, Fab Labs want to introduce a bio area. Both movements are rapidly growing. Once our PCR machine is validated and known, we are planning to reach medical labs.
Our vision is to create accessible tools that enable the diffusion and overall sharing of biotech potential outside the major research centers and make it available to everyone.
See you in San Francisco!